They are coming here. Okay. Okay, so good afternoon and good morning to the other side of the ocean. So it is a pleasure to introduce Jeff Gore. Um, Jeff Gore is an associate professor of physics at the Massachusetts Institute of Technology, MIT, in Boston. He studied at MIT in the late 90s as an undergraduate, and after receiving his PhD from University of California, Berkeley in 2005, he established his group at the MIT. His PhD focused on single molecule biophysics, and his lab is doing extraordinary research on systems and synthetic biology. They are combining experimental and also theoretical and computational approaches. And these include works about competition in bacteria, network structure of microbial metabolism, cooperation, cheating, microbial resistance, and also collapse in biological populations. So I would like to highlight a work that I really like. It's a work in 2012 about yeast, in which they were able to build a saddle node bifurcation diagram experimentally. And they are those also interested in tipping points and transitions. Okay, Jeff, thanks for accepting our invitation. As I said, it is a pleasure, and you can start whenever you are ready. Thank you so much.
All right, well, first, thank you very much for both the introduction and the invitation to speak here and to have the chance to tell you a bit about some of the ideas that we've been interested in recently. So let me just share my screen. I'll confirm that everybody can see it, and then we'll get going. Does that look okay?
Yeah, that looks great.
And also, I want to encourage you, if you'd like, if you have any questions, anything's not clear as we go, you can feel free to ask a question in the chat. Actually, one of the collaborators on this project is logged on, and hopefully he has the ability to respond to the questions as we go. But if we don't get to it during the talk, I can always answer it at the end. So,
yeah, hopefully I can answer. I'll try the best.
that's Danny. All right. Great. So what I want to do today is tell you about a recent set of ideas that we've been excited about in my group, which is basically trying to understand, what, if any, kind of universal dynamics are present within complex multi-species microbial communities. Right. And just as kind of starting background, of course, we all know that that microbial communities are tremendously complex. First, they're complex in terms of just the number of species that are present. So this is a false colored EM image of the human gut microbiome. So basically, this is an electron microscope looking at a little bit of poop. And what you can see is that there's a tremendous diversity in terms of the number of different kinds of cells that are present in here. And we think that this diversity is really essential to determining the function of this and many other microbial communities. Now, in addition to this species level diversity, there's also tremendous diversity in terms of just the range of different kinds of interactions that are present between the different community members. Right, so if we could imagine that we had the ability to see how it was that these different species or cells were interacting, then what we might see is that first, some species are interacting via resource competition, so they're competing for different things to eat. Other species might have other negative kinds of interactions like predation or the production of toxins that are inhibiting other cells. Of course, much of what we and others are thinking about these days is not just these sorts of negative interactions, but also a variety of positive interactions. Right? So there can be, for example, cross-feeding, where metabolic byproducts leak from one cell and can be consumed by other cells. There can maybe be cross-protection against some sort of environmental stress. And from the standpoint of modeling and theory, in many cases, what we want to do is we want to compare the strength of these sorts of interspecies interactions with the strength of intraspecies interactions. Because even two cells of the same species will still be interacting. They'll still be competing, for example, for the same food to eat. Right. So already this slide is getting kind of full, and it's and it's hard to make sense of what's going on. But things could be even more complex, because in addition to these sorts of purely pairwise interactions, there can also be a variety of what you might call higher order interactions, whereby the presence, say, of one species, this yellow species, could fundamentally and really qualitatively change the nature of the interaction between two other species. So what we're imagining here is a case where this yellow species maybe produces an enzyme that degrades the toxin that was produced by that blue species, right? So this is really a case where you can't describe the interactions just by the pairwise presence of species, but also you need to know about 3 or 4 other species that are present there. All right, so looking at this sort of potential interactions, it's easy to kind of maybe be overwhelmed and just kind of give up and say, all right, well, there's no hope in really predicting these communities in any way. And moreover, and certainly you wouldn't expect any sort of universal behavior within these sorts of complex communities. Okay. On the other hand, we know that a variety of other systems that you might describe as being complex systems do actually display surprisingly simple emergent behavior that can be summarized through just a few key variables. Right? So the kinds of things that we learn in high school are, for example, the pressure of a gas is described by just a few simple things, right? You want to know the number of molecules that are present, the temperature that those molecules are at, the size of the container. And actually, those things do a good job of telling you about the pressure, even if there's multiple different species of molecules present within the gas. And even if there are interactions, even if it's not an ideal gas, you can still have corrections, right? There's the van der Waals formula to tell you about, all right, volume exclusion and so forth, right? So actually, in this case, you can actually quantitatively predict the pressure of this gas, even for relatively complex mixtures of molecules. On the other hand, these molecules are weakly interacting. So you might say, all right, well, that's a little bit of a too easy of a problem. On the other hand, there's also things like the viscosity of a liquid, where the molecules in honey, for example, are really quite strongly interacting. It's a complex mixture. But nonetheless, what emerges is this, say, the single number that tells you really quantitatively how honey is going to flow as you tip it over above your breakfast plate. Okay. So it's really a simple emergent description at a higher scale that comes about as a result of the interactions between the molecules at the lower scale. Right. So these are the kinds of things that I would describe as tremendous successes that come from the physics of studying these gases and these liquids. And what we'd like is to ask, are there any circumstances in which maybe something simple emerges from complex, in our case, microbial communities? And I want to just push this analogy one step further, which is that once you have such a simple description of what's going on, then what you would really like, and what's been tremendously powerful, is to summarize our understanding through a phase diagram. Right. So in the case of, for example, many materials, and here we're looking at the phase diagram for water, what the fundamental insight here is that through just a few key variables, in this case, temperature and pressure, it allows you to really make a striking summary of the behavior of the water across this big range of conditions. Right. Indeed, if you have a cup of water at, say, room temperature, room pressure, what we know is that we can actually boil it. We can make it go into the gas phase by either increasing the temperature or by decreasing the pressure. Right. All right. So these are the kinds of things that this is the kind of understanding that we have come to really appreciate within physical sciences. And what we would like is some, at least, tentative steps in that direction within the field of community dynamics. All right, so what I want to do today is tell you about 3 different aspects of potentially universal behavior present within these multi-species communities. In particular, we're going to be studying them in multi-species microbial communities. All right, so first I'll tell you about some universal patterns that are predicted based on some simple theoretical models, like this generalized Lotka-Volterra model, which predicts that there are universal patterns of first extinction and then the loss of stability that leads to persistent fluctuations within the community. Right? And in particular, what is expected is that there's going to be a loss of species and stability that's observed as you either increase the community diversity or the interaction strength. So those two quantities are going to be our key variables that we're going to study both theoretically and experimentally. All right, so this is in the context of our phase diagram. These are our temperature and pressure, if you like. And then finally, I'll tell you about some recent experiments that we've been doing motivated by recent theory, which explores this question about the effect of dispersal between different communities. In particular, we'll show you, I'll show you some recent experiments confirming theoretical predictions that low-level dispersal between different communities can maintain diversity over the long term across this so-called meta-community, the community of communities. All right, so let's get started. So just as our group is an experimental group, but I'd say I'm an experimental physicist. So what I like to do is get excited by the theory that other people are doing, probing some general conceptual idea that I think is really exciting, and then I use that to kind of guide some experiments in the laboratory that we hope will shed new insight into these theoretical or conceptual ideas. So what I'm going to do here is just show you the results of some simulations that we have done, guided by recent theory. And this work, actually everything I'm going to be telling you today is in collaboration with two talented theorists, Guy Boonen at Technion in Israel, and Matthew Barbier, who is a theorist in France. All right, so so what we're going to do to guide our expectations is think about the dynamics within this so-called generalized Lotka-Volterra competition model. Although I'll stress that actually the kinds of things that we're going to be showing are actually quite robust to a lot of these underlying assumptions. But this is a nice and simple model. All right, so in this model, basically we look at the population sizes of each of the I species and how it changes. Excuse me, let me just get some coffee. All right, and the changes in the population sizes are going to be driven just by a few key things. So first, there's going to be the per capita growth rate is going to be maximal on its own, and then it's going to decrease as a function of both itself, right? So each species will saturate logistically by itself, as well as will be a linear function of the abundances of all the other species present within the community. So these are the interspecies interactions described by some alpha matrix. All right, and what we're going to be assuming for today is just that this alpha matrix, the effects of the species on each other, is just determined by, it's just randomly sampled across a uniform distribution, but it actually, the exact form of that random distribution doesn't matter for the purposes of what we're going to be talking about. All right, so there's going to be some randomly sampled interactions between the different species. And in addition, there's some dispersal. So there's going to be a source community with S species, right? And low-level dispersal to our focal community that we're going to be studying, right? And it's in that focal community that we're going to be focusing all of our attention, right? But importantly, we can just control the number of S species that are arriving from the outside world at some low rate. Okay. All right, so what we want to do first is just understand what happens within this model, like what are the key features, and then can we see those within experimental communities? All right, so the first thing that comes out of these models is that there's a prediction that as you add the number of species S in our source community, you'll go through basically 3 distinct phases. All right, so first, what I'm showing is these are the abundances of each species as a function of time for just a small number of species, in this case 3, right? And this is holding the strength of interspecies interactions constant, right? And so first, for a relatively for a simple world where there's only 3 species out there, what you expect is that as they arrive and they just are able to coexist with each other and in a stable fashion. Okay, so small number of species you expect here to be able to coexist. But as you increase the number of species S, then what you expect is that only a subset of the species will survive. So here we have a world of intermediate complexity, 12 species. They're arriving into our focal community. And what you see is that some species here are surviving and some are going to very, very low densities. And indeed, those low species abundances, they're only above 0 because of that dispersal. So they're arriving and then each individual is dying and those species would be going extinct, except that they just keep on arriving at some low rate. So they're present at very low abundances. So but that's below a threshold that we're gonna call extinct. All right, they're only being sustained by that low level of dispersal. Okay. However, the species that survive, which is some subset of the initial 12, they survive in a stable fashion. Okay. Then finally, as we increase the complexity of the world yet further, here with 50 species, you see a qualitatively different regime, where now it's again the case that only some species are surviving, and those that survive persist through these persistent fluctuations, which eventually you can prove are chaotic. Okay, so these are kind of the 3 phases that you expect within this model as you increase the diversity of that community. And indeed, you actually see a very similar set of transitions if instead of increasing the number of species S, you instead increase the strength of interspecies interactions. And so I'm just going to show you now what happens if we hold the number of species constant at 50, and we're going to go from very weak interactions between the species, and these are all inhibitory interactions. And we're going to kind of just dial up the strength of those interspecies inhibitions. All right, so very weak interactions, the species survive together in a state that is again stable, so all the abundances are constant over time. But as we kind of start dialing up the strength of interspecies interactions, what you see is that you see something similar to what we had before, which is now some of the species are surviving, some are going extinct, they're going below that threshold, but the ones that are surviving are coexisting in a stable fashion. Okay. Finally, as we increase that interaction strength yet further, right, this is the mean of the random distribution that we're sampling from. What you see is, again, something that looks rather similar to what we had before, which is that some of the species survive, some go extinct. The ones that survive are surviving through persistent fluctuations that are eventually chaotic. Okay. So you see is that there's something rather similar as we increase the diversity of the community in the outside world that we're dispersing from, or if we increase the strength of interspecies interactions. All right, so given that, it's tempting then to kind of think about this in the context of a phase diagram, where what we're going to do is we're going to look at basically the stability and the survival of species as a function of these 2 key parameters. Right, so this is the initial richness, S, on the x-axis, and the mean interaction strength on the y-axis. Right, and what's being plotted is basically the fraction of the communities that are fluctuating. Okay, so what you have here is that there's basically a few different regimes. Down here at the bottom left, you have a community that's at a stable equilibrium. But then as you kind of go up in interaction strength, or as you increase the richness, you arrive at a situation where basically all the communities are displaying persistent fluctuations. The other thing that you might be interested in plotting would be the fraction of those S species that are able to survive within that focal community. Right, so that's what I'm showing you here. Again, the same axes, so richness S, interaction strength. And what you see is that, again, at the bottom left, you have a state where all those species are surviving. So the fraction that survive is 1. Then you go through a regime of partial coexistence, where species start going extinct. And then you cross and this solid line is the same in both. Then you cross this point where you get a loss of stability, where you start getting fluctuations, and then the fluctuations grow, the fraction of the communities that are fluctuating grow larger and larger. I think that one thing that is interesting to note is that if you look at the fraction of the species that are surviving here on the right, as you go further and further into this fluctuation regime, nothing dramatic appears to be happening in terms of species, right? So in particular, as you get more and more fluctuations, right, you see there's a big change over in the color on the left. There's not a dramatic change in the fraction of the species that are surviving. So as you enter this fluctuating regime, you do not experience a dramatic loss of species in terms of the fraction of species that can coexist. All right, so these are the basic expectations from some of these simple models, right? And so the question is, can we actually see signatures of this potentially universal behavior within experimental communities? Okay. All right, and if there are any questions now, I'm happy to take them, or I can show you the experiments.
Yeah, I have a question, Jeff. Sorry. So, have you checked if in this model you may have some kind of bistability, because then you would have some kind of dependence on the initial condition? Thank you.
Right. So within this regime, we do not. As you increase the interaction strength, then you eventually get alternative stable states and so forth, right? So yeah. So as you come up here,
Okay.
Go up, yeah. And actually, there's other ways of plotting this phase diagram that Guy Boone and Matthew Barbier have done, where you go into where this is more clear in terms of the alternative stable state regime. Yeah. Okay. Right. So let's think about our experiments. All right. So what we're going to do is take basically 48 different bacterial isolates, okay. And what we're going to do is we're going to choose different random subsets, S of them, and we're going to use that to inoculate into a well-mixed batch culture. So what that means is that we're going to pick S out of the 48 species. We're going to start it growing together in a controlled experimental kind of setup. And we're going to do daily growth dilution cycles where we allow these species to grow together. They're going to be competing for the resources. They're going to be secreting whatever they want to secrete. Then at the end of the day, we perform a dilution where we transfer over just a few % of the cells into fresh media. And together with that, we disperse, that we take, again, very low level of those same S species at the kind of 10 to the -4 level to re-inoculate into that community. And then what we're going to do is basically let this go for 10, 12 days, and then look to see what the dynamics are by doing 16S sequencing. Okay. And key things are we're going to be varying the number of S species, and then later we're also going to be changing the media to control the strength of interspecies interactions. Okay. All right, so let's just start by thinking about this phase diagram. What is it that we might be looking for experimentally? Well, first, what we're going to be doing is controlling the number of those S species, right? So what you expect is that maybe for a small number of species S, you will be in this blue regime where only a small fraction of the communities are fluctuating. But as you add species S, then you expect to get more and more fluctuations within that community. Right, so the first most obvious thing that you can look at is just look at the biomass of those communities over time. Right, so what I'm going to be doing is showing you, different subsets S for starting from 3 going up to 48, and looking at just biomass as a function of time. Right, so here each of these lines, there are different 3 species sampled out of that 48. Looking at biomass as measured just by the OD, if that means anything to you, as a function of time, what you can see is that over here, most of those communities have stable biomasses over time. But here, actually, one of those communities is displaying persistent fluctuations in biomass. Right? And indeed, we also have done sequencing of these communities, and we found that the biomass fluctuations are also consistent with abundance fluctuations. Okay. In the sense that if the biomass is fluctuating, then the species abundance fractions are also fluctuating. Right. So here, most of those communities are stable, but one of them is fluctuating. As we go to larger and larger S, so as we go to more complex communities, what you see is that a larger and larger fraction of those communities are displaying persistent fluctuations. And this is in terms of biomass, and also those were all also showing fluctuations in species abundance as quantified by 16S sequencing. All right, so what this is saying is that it seems like this expectation that you get from the simple model actually seems to bear out within experimental communities, where if you just plot the fraction of those communities that are fluctuating as a function of that initial richness S, what you see is that for very simple communities, and actually the 1 and 2 species communities in these conditions didn't display any fluctuations in biomass or in abundance. But as we get to more and more complex communities, then we kind of are traveling here to the right, where we get more and more of those communities fluctuating, where the communities, say with 24 species, almost all of them are displaying persistent fluctuations. All right, and just to show you some examples of the species abundances as a function of time in a few of these different communities. These are examples of the 3 species community that was moving around a lot in terms of fluctuations as compared to equilibrium community, and same thing for 6 and 12. All right, so this is just to show you that we have actually done the 16S sequencing for these communities, and we see that indeed the stable biomass communities are displaying stable abundances, whereas the fluctuating ones are also fluctuating in abundances. All right. There's another interesting thing that comes out of this theoretical analysis, which is that there is an expectation from theory that there's a sense in which these persistent fluctuations are actually enhancing community diversity. All right, so what I'm showing first is the fraction of the species that are surviving as a function of initial richness S. The mean is just in blue. But what you see is that different individual communities display widely varying survival fractions, even for the same S. Right. So if we look here at say the 24 species kind of communities, what you see is that some of them survival fraction here is below 10%, whereas others survival fraction is up here, 2/3 of the species survive. Tremendous diversity just for different random samplings of that alpha matrix. Or what's interesting is that among these different individual communities that with different alpha matrices, what you see is that the communities that actually display persistent fluctuations are the communities where many more of those species are surviving. Right? So even though we often think about chaotic fluctuations as being really bad, because abundances can go really low, you can get extinction and so forth, here you can see that there's a really, I think, interesting sense in which the fluctuations are actually enhancing diversity, because it's really the communities that are displaying fluctuations that are much more diverse. And this is all holding that initial S constant. Right. All right, so the question is, can we see anything like this in the experiments? And of course, we have much it's much harder to collect experimental data than simulated data, but it seems like this pattern is also holding within the experiments, right? Where if you look at, say, our 6 or 12 species communities where we have many different combinations, what you can, and again, for us, resampling the alpha matrix is equivalent to sampling which of the 48 species we're choosing, right? So these 6 species communities, we choose any individual 6 out of the 48, and they're going to have different interactions amongst themselves, right? So what you can see here is that these stable communities here have much less diversity than the communities that are fluctuating. That's true, and then you can really see it in the 6 and the 12 pretty clearly. Right? So this is really saying that you expect from theory, and it seems to play out in the experiments, that these persistent fluctuations are somehow enhancing increasing the diversity that can be maintained within the community. Right? And of course, the causality maybe goes both ways. So you have to figure out how to think about it. But I think this is to me a kind of a surprising feature of the model and of the experiments. Okay. All right. So so far what we've done is we've just for a fixed kind of distribution of alphas, right? So this is saying, all right, we're taking all that complexity of interspecies interaction. We're saying, all right, there's going to be some distribution, right? We don't know what it is, right? But there's going to be some distribution of interactions between the species that we're sampling from, depending on which species we choose. Right? And the question is there any way that we can vary that distribution, vary the distribution of interactions between the species in order to probe a little bit more of this full phase diagram as predicted by theory? Right? And the nice thing is that there is recent that a postdoc in my group, Christoph Rotsky, recently demonstrated that in this media environment that we have, we can vary the mean interaction strength by changing resource concentration. And the basic insight here is that although we often think about species interactions as being these direct interactions, right? So we draw arrows between species. This is not really the way that it happens. Really what happens is that for most of the interactions between these microbial species, each species has some metabolic capabilities in some environment. So it's going to grow in some way. It's going to be eating things from the environment. It's going to be secreting things back to the environment. But it's really metabolic growth leads to changes to some shared chemical environment. And indeed, in this environment, what we have found is that as you increase the nutrient concentration, you get more growth and you get more chemical modifications to the environment and stronger interactions between the species. And this is nice because this provides an experimental platform for us to change this mean interaction strength. So basically what we had found before is that we can increase the nutrient concentration in order to move up in this kind of phase diagram. All right. So the question is, can we actually take slices of this phase diagram by performing this experiment at multiple nutrient concentrations? All right. And in all right. So yeah. So let's look at it. All right. So this is now we're gonna take a slice where we're gonna look at multiple combinations of 24 species out of the 48 at low nutrient concentrations, then going up to higher nutrient concentrations, which is what we had before. Right. And what you can see here is that in low nutrient concentrations, all those different combinations of 24 species, they're all displaying stable biomass. And indeed, they have stable abundances as well. As we increase the nutrient concentration, however, what we can see is that indeed we can kind of go, experimentally to this regime where most of those communities are displaying persistent fluctuations. Right, so this is consistent with what you expected from theory. Right, so we can kind of move up that phase diagram. All right, so this is great because now we have the power to kind of move left, move right, move up, move down on this phase diagram. And so I'm just going to show you some data where we have kind of varied across S. So we're taking 3 different slices on high nutrients, intermediate, and low. And then we can just ask, well, what fraction of those communities are displaying persistent fluctuations, right? And what you expect from the model is that basically the transition from stable to unstable or oscillatory communities that'll happen at larger S as we go to weaker interaction strength, right? And indeed, that seems to be precisely what we see experimentally, right? So the blue data up on top is what I showed you before for the onset of persistent fluctuations in high nutrients. But as we go to intermediate nutrients, we still get this onset of fluctuations, but it occurs at larger S. Whereas at low nutrients, we see no fluctuations, even up to our full 48 species combination. Of course, we only have one of those combinations because there's only one way to pick 48 species out of 48 species. Right? All right, so this is saying that this phase diagram that I showed you at the beginning of the fluctuations actually is providing significant guidance regarding what happens within actual experimental communities. And finally, it is maybe also fun to just plot the fraction of the species that are surviving as a function of S for these different nutrient concentrations. And what you expect here is that basically for low nutrients, as you increase S, you're, of course, as you increase the number of species in your community or in your world, a smaller fraction are going to be able to survive. But this effect you expect to kind of saturate, because this blue region is actually a surprisingly flat plateau within this phase diagram, right? And that's why this maroon and blue curves are so similar to each other, because this is a nearly flat kind of plateau up on the top right. And indeed, that's actually what we see experimentally, is that as we increase the interaction strength, we have a smaller fraction of species surviving, but that effect kind of saturates, in that our intermediate and our high nutrient conditions display very similar fractions of the species surviving. And I want to note that that's true despite the fact that if you look at the dynamics here on the bottom right, they're actually displaying significantly different dynamics in terms of stability and fluctuations, right? The blue and the maroon data are not on top of each other in terms of the fluctuation dynamics, but they actually are roughly on top of each other in terms of the fraction of species that are surviving. And that's kind of what I alluded to before, which is that you are losing stability, you're getting fluctuations, but you're not changing very much the fraction of species that survive. And that was true in the model, but it also seems to be true within these experimental communities. All right, so yeah, and so it seems like these emergent fluctuations are maintaining high diversity, and that's by allowing local blooms of rare species. All right, so here at the end, I just want to look at the time to make sure I'm not. All right, great. All right, so now to just kind of taking a little bit of a step back, what we were interested in is asking what would be kind of universal dynamics within a focal community as a function of some key variables that focal community might be kind of dominated by. And what we found, or what we've been arguing, is that these two parameters, this species diversity S and the mean interaction strength, can provide a great deal of insight into what's going to happen within this community, right? The fraction of species that are going to survive together, and whether that community survives via, goes to some stable state or displays persistent fluctuations. Of course, we've a little bit swept under the rug where those S species come from, right? And in some ways we've been thinking about, all right, you could say, all right, well, here, I have some gut community, and its dynamics are determined by, all right, there's some number of S species that I'm going to be exposed to in the world, but I didn't say anything about what determines that S, right? I just said, all right, we're going to allow that S to vary and ask what happens. But we know that actually it's challenging to have large numbers of species S survive together, because there's, again, this chicken and egg problem, right? It's like, oh, well, how is it that they survived out there if they can't survive here, right? However, there's recent theory that has been arguing that basically one way to get those large number of S species to survive together in the outside world is that maybe we should not think about the outside world as like a single big community where it's hard to get large numbers of species surviving together, but instead you should think about the outside world as being, in ecology, we often talk about this as a mainland, because this is from the theory of island biogeography. But you can think about this outside mainland world as a meta-community, where it's a community of communities, right? So you have many different patches that are each connected to each other by some amount of dispersal. Now, in this case, you can ask, well, how many species are able to survive within this meta community? And you compare that to the number of species that could survive within a single community. And what's been shown in some recent work by both Daniel Fisher as well as Guy Boonen is that actually you can get many more species surviving within the meta community than could survive within an individual community, and that this is a very strong function of that migration or dispersal rate, the strength of the connections between the different patches within that meta community. Right? And basically, in the at very low migration or dispersal rates, you basically have disconnected communities, and then you can get local extinctions. Whereas at very high migration or dispersal rates, then those communities are all very strongly coupled, and then they actually start all fluctuating together, and then you get extinctions, right? However, there's an intermediate regime where you get many more species surviving across the meta-community than you could within any individual community, right? So there's a peak in the diversity as a function of this dispersal rate, or the strength of the connections between the different patches within the community. Okay. So I think that this is a really interesting theoretical prediction, and it motivated us to try to see if we can observe this in our experimental system. All right, so how is it that we're going to try to do this? All right, so this is what I showed you before for the experimental platform to try to understand the phase diagram within the focal community. All right, now what we're going to do here is modify a few things. All right, so first we're going to inoculate, instead of using defined isolates of, say, some number, 48 species. What we actually did for this experiment is we just took soil from the front yard of the laboratory. We just took some soil and we vortex, we mix it up, and we just inoculate with that full community, right? With lots of complexity, right? So this is a way of just starting with a lot of many different species, right? So we inoculate from soil, and instead of, again, here just inoculating into a single well, what we do is we inoculate into, actually in this case, 8 different wells, right? So the 8 wells are going to be our meta community, right? So we have 8 different patches, right? 8 different wells that we're going to be studying. And then finally, instead of here, although in the previous experiment, what we did is we had a continual low level of dispersal from that source community. Here, what we're going to do is we're only going to inoculate that first day, right? So no dispersal from the source community, because what we wanted to ask is, can we maintain diversity within the community without having dispersal from the outside world? Because here now we have a meta community, so this is the entire world, right? However, what we want to do is, we have these 8 wells where we inoculated them the first day. All right, what we're going to do, instead of having dispersal from the outside world now we just have dispersal where we just have a low-level dispersal between the 8 different patches, or the 8 different wells within our experiment. All right, so I'm only showing 2, but there's actually 8. Right, in theory, 8 is actually sufficient to lead to this effect. Okay. So that's the basic design of the experiment. Right, so then you can ask, well, how much diversity do we observe within that meta community, the 8 wells, as a function of the strength of the dispersal between them? And is there any peak? All right, that's the basic question. And indeed, it seems that this is preliminary data that we're just studying now. But indeed, it looks like there is a peak in the diversity, or the richness, the number of species that were able to coexist within this meta community as a function of this dispersal rate, right? And what you can see is that basically at low dispersal, we observe about 10 species coexisting within that meta community, whereas the diversity more than doubles as we increase the dispersal rate. Right, but it's not just that dispersal is good here, because again, there's a peak in the diversity as a function of the dispersal, where as we have strong dispersal, strong connections between those different patches, the diversity actually comes down lower than what we saw at low dispersal. Right. Now, there's a lot of interesting things you can do with the actual sequencing data in terms of the dynamics and so forth. This next slide is a bit much, so I'm just gonna, warn you. All right, so this is what I'm gonna show you is the species abundances across each of the 8 different patches or wells, and that's gonna be as a function of time. And this is just for one of our meta communities, and then, of course, we did multiple of them. All right, but let me show you what it looks like. All right, so this is one meta community that's composed of 8 different communities. All right, and these communities are, again, connected by low-level dispersal. And it's color-coded by which species is present there. And there's a few things that you can say. First, you can see there's a lot of fluctuations, right, as expected. And indeed, that's the basic mechanism by which this was predicted to happen. right? So there's persistent fluctuations. Second, there's diversity between the different communities in the sense that the different communities are behaving differently. And that is, again, predicted by theory. And that's, again, part of the mechanism by which the global diversity is maintained, right? So you can see that if you look, some of the communities look rather similar. So here, community 1 and community 5 look rather similar. Community 3 and community 4, kind of similar. But the different communities are doing different things at different times. And there are local blooms of species that take place that you can observe in one community that are not present within other communities at the same time, but may allow for reseeding of species in other communities as they go extinct. Right. So there's a bunch of things that you could ask and say about these dynamics, but I just want to show you that we indeed are probing these community dynamics and how they're coupled across this meta-communities of function of time. But emerging from all of these complex dynamics is this statement, which is that global diversity can be maintained by dispersal within a meta-community. Okay. All right, with that, I'm going to summarize so that you can ask questions. All right, so first, we're excited by this idea that there are some emergent properties that are leading to kind of universal behavior within these complex communities. And this is predicted by theory, and it seems to be borne out within experiments. And in particular, what we found is that these communities are losing stability. Well, they're losing species and then losing stability when we add species or nutrients, right? So there's a few kind of phases that these communities are in, and there's a particular order that things happen, and that is the same order whether you're adding species or nutrients here. And also what we've seen is there's this fascinating role of persistent and potentially chaotic fluctuations that are allowing for higher species diversity. And there's one sense in which we saw this within the single community, where at fixed S, the communities that have a lot of diversity have fluctuations. And then this latter part that I showed you, this is within a meta community where those persistent fluctuations are occurring asynchronously at different patches. And because they're connected by low-level dispersal, we are able to maintain community or meta-community diversity over the long term. So with that, I'm going to thank the group. So first, both of these projects are being led by Jilong Hu, a talented graduate student within the group, and he has been aided by an exceptional postdoc, Daniel Moore, who is here, and we can wave to him. And those two group members within my group here at MIT have been aided greatly by our collaborations with Guy Boone and Matthew Barbier, who are really interesting theorists working in this area and that have provided a great deal of insight for us. All right, so with that, I'll quit, and I am happy to take any questions.
Okay, thank you so much, Jeff. Your talk was extremely impressive to me as a biologist working in dynamical systems. So you are really doing very, very interesting things. So if you don't mind, I can read some of the questions that we have in the chat. So the first one is by Sasha Remina. She's asking, do you think that the organisms we currently can't culture in the lab, often because they need larger, more complex microbial communities to survive, would show similar community dynamics, particularly in terms of fluctuations in species abundance?
Yeah, right. So these are, this is a really, interesting and important question. So, we obviously, we're laboratory experimentalists primarily, and that allows us to have some degree of control over our community so that we can do things like vary S and vary the alpha, distribution. But of course, if we want to study things, microbial growth in the lab, then we can only study the species that we can grow in the lab, right? And this is, and there are many reasons that we and others cannot necessarily isolate everything from, say, the soil and grow it in the lab. One reason is pointed to by Sasha, which is that maybe the species that you see in the environment are they require the other species present in that environment in order to grow, right? So the way that we isolate is that they have to be able to grow on their own, right? I will maybe just note that actually that second experiment that I showed you, where we just inoculated from the soil, in principle is not subject to that constraint, because we did not start with isolates, right? We just started from the soil, right? So so if there were any species that were present in the soil that required other species present in the soil to grow in our lab, actually they could have persisted, right? So I think that's part of why we actually do some inoculations just from natural communities. All right, now to actually try to answer her question, I of course don't know what would happen with the species that we can't grow in the lab because we can't grow them. But I would say that given the nature of the kind of questions that we're asking about kind of these universal or potentially universal behaviors, I guess I'm hopeful that many of the dynamics that we see don't necessarily depend on maybe it doesn't even depend on whether it was soil species, or maybe that we would have seen similar things if we had done used marine isolates and done those sorts of experiments. But I of course we haven't done those experiments yet, so we don't know if that's true.
Okay, so Daniela Moore has just answered the question of Josep Mercadal in the chat. So Blai Vidiella is asking that, following the theory, what do you expect to happen if there is a fusion of two major communities, especially in your experiments?
Yeah, so I'm very interested in these questions of community coalescence. So you can take two natural communities and do that. We've been actually starting by trying to answer this question in the context of a very simple situation, which is if you take simple communities and you do this coalescence, like what happens? Does the final community look more like one or more like the other? And actually, the way that we designed that experiment that I showed you at the beginning, right? I told you that there were different combinations of 3, different combinations of 6, 12, 24, and 48. The way that we actually designed those communities was via this so-called tournament structure, where the different combinations of 3 actually were the same as the combinations of 6 and the same as the combinations of 12. So we designed that experiment to be able to address some of this question of community coalescence that you're talking about. But we haven't, we've been trying to analyze the data first in terms of this framework. And so we haven't yet figured out exactly what's happening in terms of community coalescence, but we actually have the data that's there. And and you can tell Jilan, you're very interested in this, because I've been pushing him to think about this, and he just hasn't gotten to it yet.
Okay, so Chris Micklem is asking, I was curious about the synthetic microbial communities. Are the constituents all seated in the same proportions? Were different ratios also experimented with?
Yeah. Okay, good. Yeah. So for this experiment, we did roughly equal abundances and only equal abundances. So we did not explicitly go after dependencies on the initial conditions. We of course have done a lot. We're very interested in alternative stable states, and so we've studied this in many different ways. And yeah, so this is something we're very interested in. But for this experiment, we did not probe that. So yeah, we really don't know what would happen.
Okay, also Chris, and are common species enriched in communities occupying set regions of the phase space? I just wonder if perhaps the sensitivity of communities to particular species in terms of their stability fluctuations can be determined.
Okay, yeah, so this is another interesting set of ideas. Yeah, so we do not, so there are common species that, I guess maybe you're thinking about common species from the species that are common in the environment, right? And then because there's also questions about species that are common once they arrive in our laboratory, in our laboratory community. And I'd say we do not know enough about the species interactions to be able to say very much. In this case, so we do a lot of things of pairwise interactions between species, but we have not characterized all the pairwise interactions among these 48 species, just because the 48 squared ends up being a lot of pairs. So we actually do not know all of the interactions between all the species in our community right now. And we've done a subset of them, and we're of course very interested in this question. But I will maybe just note that I think part of what I'm excited about with this approach is that we can actually make predictions about things like fractional species that are going to survive and the probability of a community being stable without knowing anything about particular identities of species or their interactions, just based on distributions of, say, the alphas. So that's part of the philosophical thing on this project. But of course, we also want to know, like, we're diving into a bunch of these things now though as well.
Okay, Nuria Conde was asking about metabolomics data to see how functions change, and Daniel already answered. Thanks, Daniel. So I have a question. I have been always fascinated by this search of theoretical results in experiments, and I don't know if you know about a concept that it's called homeochaos, that was discussed in the early 90s by Kaneko and Ikegami. Homeochaos is a type of chaotic behavior in which you have, let's say, a lot of positive Lyapunov exponents, but these Lyapunov exponents are very close to zero. So it's a kind of high dimensional hyperchaos. And under these dynamics, what you have is that you have chaotic fluctuations, but the fluctuations are very narrow. So this is a kind of very stable dynamics in an unstable set as a chaotic attractor. So I was wondering if, I guess that in the experiments, perhaps it might be difficult to work with the time series data analysis, but perhaps in the model, you could try to find this homeochaos by computing the Lyapunov exponents, but because then, for example, as many species you may have, one might expect for some parameter values of this alpha yj to see a kind of slight increase in the survival fraction of the population. So I don't know if this might be an easy thing to identify in the lab, but
so yeah, there are a lot of things. Yeah, so I don't know how our chaos would likely be, how it would be related to chaos. And in our experiments, we haven't demonstrated anything chaotic. So I want to be clear about that. But it's at least model, you can demonstrate this. I'll also maybe just note that this issue of the fraction of species that survive is a subtle one, and that's true particularly in the chaotic regime, because the longer that you wait in terms of asking, is a species present at high abundance, the larger your window, the more species that are going to be present, right? In the chaotic regime. If it's stable, then that's not true, because it doesn't matter how long you're looking at it. Whereas in the chaotic regime, if you wait long enough, eventually everything's all going to show up, right? So then it's actually a surprisingly subtle issue, from the theoretical perspective. Things are simpler from an experimental perspective, because we look at the end of 10 days and ask who's there above threshold, right?
Sure. Okay. Thanks. So any more questions? You can use the chat or turn your microphone on. Okay, so in principle, we do not have more questions. So thanks so much, Jeff. I think people really enjoyed your talk. Thanks so much for being here with us. Okay. So we will now move to the second speaker, Miki Ebisuya. Okay, so we move to the last speaker of today's session on systems and synthetic biology and dynamics. Nuria, I don't know if you can start sharing.
Yes, I think.
Okay, so next speaker is Nuria Conde. Nuria is a postdoctoral researcher at the Complex Systems Lab at the Universitat Pompeu Fabra and the Institute for Evolutionary Biology, which is placed at the PRBB in Barcelona. She holds a major in biology and in computer science and has performed her research thesis about biocomputation. Actually, today's Eulalia de Nadal talk was explaining some of her PhD research. Nuria teaches biology for architects, artists, and designers, and is a founder member of the Do-It-Yourself Bio Barcelona, the first biohacking group of Spain. And her expertise is on synthetic biology and distributed biological computation. And she also worked in synthetic lethality in cancer, synthetic collective intelligence and in complex networks theory. Okay, Nuria, so you can start whenever you want. Thanks.
Thank you, Josep. So I'm actually very excited today because I'm going to present the fresh, super new work of our group that is still, it will be coming soon in press. It's the result of a collaboration between our group, the Complex System Lab of the UPF, with Ricard, Blay, myself, and the students, but also with collaboration of the Dr. Sardañez and Guillamon from the Center of the Recerca Matemática. Let me start, remember or introducing you the concept of self-organized criticality. The self-organized criticality is a property of a complex system that's able to be attracted by itself, to organize itself around the critical state. So the system tends to go to around the transition between two different regimes. This behavior is supposed to be robust and scale invariant. The archetypical example that I think it explains it very well and it's easy to understand is the sand pile that was proposed by Bach, Tank, and Weisenfer. It's just like, as you can see in the picture, you have a pile of sand and then you have a ratio in which you are like adding more sand grains or rice grains. And then the angle of this pile is like increasing and increasing, since in a moment that you will have an avalanche of all the sand. And then you can restart again. You keep adding grains of salt, and then again, the angle will increase, and then so on and so forth. What if it was demonstrated, actually the experiment that they proposed was not actual sand, it was a lattice in cellular automata, but what was seen and demonstrated is like all the system is posed in this critical state, and that exhibits a power law scaling with the size of the events and the duration. So you cannot predict easily when the next avalanche will come or the size of this one. There is not this property, the self-organized property is actually a property that is exhibited in many systems, because any system that's in non-equilibrium systems and have many degrees of freeness and exhibits nonlinear dynamics, it can be susceptible of exhibiting self-organized criticality. It has been demonstrated also in spiking neurons, in solar flares, earthquakes, financial markets, burst of activity in ant colonies, forest fire initiation and propagation, urban densification, so on and so forth. As I already explained it for the sand pile, all these systems that have this self-organized criticality, you can detect them because they will exhibit this power law scaling of the events. Here I'm showing some of the, because you don't know to believe me that this is very well known, and you can see how if you take, for example, earthquakes or solar flares, and you plot the size of the events, of the intensity of the earthquakes, of the solar flares against the complementary cumulative probability distribution, the power law, it's clearly seen in the graph. And also, and this is very impressive, at least for me, it doesn't matter which paper you take or which measure they decided to do in the network of neurons. If you take the spike cluster size and you plot it again with the distribution of the size of the neural avalanches, you can see again that you have a power law scale, and that this exponent is around 2. It's like the magical number that goes between 2 and 3. So the question that arises in our group is how we can make all this self-organized criticality inside of a cell. Well, a good starting point is to use this super simple model that we already have for free. So our starting point is a promoter with a high ratio of expression with mu, and then you have this protein that has to be tagged to in order to be degraded for a complex of degradation. We choose to use the clip X and P protease complex. It's known that in a culture of E. coli that it's in exponential phase, it's a constant. There is a constant level of clip X and clips P. It's around not many molecules. They say that's around 100 for each cell, and that they can degrade 10 to 5 molecules of GFP per cell, target GFP per cell and per replication cycle. It seems a lot, so they are very active, but since they are not so many and they have so many other proteins to degrade, it's actually very easy exist a lot of promoters that are able to produce enough proteins to actually saturate and congest the system of proteolysis. If you do that, that means that there will be a level of activation of the protein, of the promoter, that will lead to a concentration of the protein that will start to congesting the system. So the system will exhibit two phases. The phase that you are producing and degrading the protein, we call that free phase. And then there is another phase when you are overproducing the target proteins, and then the proteolytic machinery is not able to degrade them, and then you will be in a saturated, or as we call it, congested phase. That was a starting point, but of course the system does not exhibit self-organized criticality, and we needed to do a little bit more, not too much more. I think that one of the beauties of our work is that it's very simple, but is still able to reproduce this self-organized criticality. Here I present you both at the same time, the genetic design and also the model design. I will keep telling both at the same time. So you have a promoter of a strain nu, that in our case, it was the PELAC-I promoter, and that was expressing a sigma-1 protein that in our design is the GFP, also target for the CLIP-X, CLIP-P protease degradation. And in the other part of the design was a promoter that in our case, we choose the PBAT promoter. This decision was done because we wanted a promoter that do not exhibit levels of leakiness, so that 0 will be mean almost 0, really, and also allowed us to have a broad range of expression. So this nu could take a broad range of sorry. This new parameter can take a lot of values. And then under the control of this promoter, this control of nu, you will have a sigma-2 protein that in our design is the repressor protein Lotka, also target for being degraded, followed by the red fluorescent protein, also target. That in the red fluorescent protein it's used for us as a proxy to know how much of our repressor protein is present inside of the cell. Actually, the repressor protein will control the amount of GFP, because the Lotka will repress this production of our order parameter or GFP. Again, we choose that repressor because it has the property that can be inhibited by the addition of IPTG intermediate, and that gives us another point to tune and explore the parameter range in the experiments. So in this slide, I think is the core of the presentation. You have the genetic design, you have the model design. I I'm happy to say that this design could be much more complicated, but we put it in these two equations, and they reproduce all the behavior. So just slowly, for all the biologists that I think will appreciate that minutes that I will take to explain that, the production of our GFP or sigma-1 protein will be, of course, produced by a ratio that it's called here Nu1, but it will be also influenced by the presence of IPTG. That's the mu that you can see in the denominator of the function of the strength of the promoter, and also will be influenced by the inhibition of the concentration of Loci or sigma-2. As well known, Loci have to dimerize to actually perform this repressive action, and it's all captured in this F sigma-2 function. And then we have an spontaneous ratio of decay of GFP and an active ratio of decay of GFP that's performed by the proteolytic machinery. The proteolytic machinery, it's coded in the last equation, tau, that it depends of sigma-2 and sigma-1. That's actually the beauty of the model, because the proteolytic machinery will degrade GFP and Loci at the same time, that are both contributing to the collapsation of the system, as you can see here in the equation. And then the other one have no more explanation. Or our abinose in the medium will increase the Nu2 parameter, and so on and so forth. So with this in mind, and with these simple equations, we tested the model. We'll talk about the results, but we have been demonstrated, and that's the point, that when you are doing synthetic biology, you are happy to see that actually exists a point for the thing that exists this point in what you have a promoter Nu1 that can have an strength with high probability will put your system in this SOC state. So in the middle of the two phases between being free and congested. If the model shows that, sorry, the model shows that the most probable value for the N1 strength, it's posed in this critical state in which the system will change from free from congested. Also, I will show you now some of stochastic simulations using the Gillespie method that we did. For me, the clear example that the model is capturing the system is that in this right panel, you can see that the inlet of the right panel, you will see like the time series of the values that our sigma-1, the GFP, will be taking in the simulations. You can see that the spikes that you have are fluctuations with not without any fixed period, and also with a very variable size that for us will be intensity of GFP. You can see, as we have been explained for the other systems, that if you plot the amount of GFP or sigma-1 against the cumulated distribution of this parameter, you will see the power law scaling that we have been talking about, and also the model shows that we are around this -2 exponent. These are the parameter sets, like we'll have when Nu2 is around T-1, that it has been shown by the model that it's where the maximum variance of fluctuations of sigma are obtained, and also where the simulation show the slow fast dynamics if you plot it in a flux vector sigma-2 against sigma-1. For the biologists, that means that you activated a little bit in the correct point the promoter for expressing the repressor. And you only will see this sock behavior that shows the power law when you also add to the medium, enough IPTG or mu to fine tune and actually activate the sock model. Otherwise, if you don't do that, you only have a Gaussian signal. These are results from the model. If you read the paper soon, you will see that they did much more than that, and they are in the audience if you have more questions. But now I will start the job of actually building that for a real cell. So you can see the model is exactly the same. So you have expression of GFP under the P-LAC-I, and then you have the expression of P-LAC-I under the PBAT promoter, and then you have the reporter protein for LAC-I, but absolutely everything it's put in the same construct, because it was very important that we got a high level of that proteins, so we can reach the saturation easily, and we can also see a good signal. But also we wanted to ensure that the population got the same amount or a number of plasmids inside of every cell that was the most uniform possible. That's why we choose the E. coli XL1 that can hold a lot of plasmids inside without recombination. And then we had to develop a method of growing the cells, diluted the next day, coming from just one cell and assuring a very uniform and in a very happy exponential phase culture of E. coli. And then we also had to go to the flow cytometer, and we have to adjust the machine because these cells are actually very, very tiny, and they have to exhibit different levels of RFP and GFP, and we wanted to be able to capture all of that. And this was a question that we received a lot from the reviewers. The population, it's the same E. coli with the same construct. I already said we tried to make it uniformly, and we did not filter the signal for anything. We just captured all the cells and we take it away, and we really found the threshold in which the debris, that's the tiny particles inside of the machine, so we just take it out the debris, but we didn't filter any cell. With this set, we were very happy the day that we could see that with our eyes. So I show you here real microscopic images of these E. coli. In the left down, you can see where a condition without arabinose and without EPTG, so there is no expression of any of our fluorescent proteins. In your left up with A, you can see what happens when you add a lot of EPTG, so you release the brake on the LAC-I promoter that's expressed in the GFP, and you have mainly, you can see few red cells, because it's not so clear that the level of PBAT of the arabinose it's tiny, but mainly all the culture was expressing a high level of GFP, uniformous high level of GFP, and the other way around. So if you check the other part of your circuit, then you don't add the APTG, and then you add the arabinose, your population will exhibit a lot of red cells, all in the same range in a high level of RFP. We assume that this also means that they are having a lot of LAC-I, and that's why the repressor of GFP was doubled. And that was the panel B in the upper right. It's what I was it took us a while to find out the point in which amount of IPTG and arabinose it's putting our system in a place, in a parameter space that allow itself to go to the shock state. And that's the second part of our job as biologists, that's going to with these uniform cultures and presenting them different concentrations and combinations of IPTG and arabinose. It might seem contradictory with the self-organized description of SOC, But the thing is that even with the rice piles, you have to fine tune and not always they exhibit sock, depends on the grind of the salt or the grind of the rice also. So what we do is look for the right parameters space, not only a given point where the sock behavior will be able to be achieved. And as you can see here, or not, these two panels that are inside of the in the gray square are the ones that are performing SOC. These are I don't know if you're familiar with that. These are the panels that just represent in the x-axis you have the red intensity for the RFP, and then in the y-axis you will have the intensity, the log in the log radio, the intensity of the GFP. And if you are only looking at the first column without the triggering of our control parameter or the repressor like I protein, you can see that in increasing amounts of IPTG allow us to move the system from the free phase where the proteolytic machinery is actually working, and the one phase in which the proteolytic state is already being starting to be congested. As you can see, the difference is between 7.5 micromolar to 10 micromolar. When you are in that state and you are having the proteolytic machinery a little bit starting the congestion, if you trigger the production of the control parameter or the repressor, then the SOC state will be achieved. And you can see that because in the Q4 panel, that's the panel in which the cells are having red, so they have our control parameter, the laci, are also exhibiting green. And if you only and also you will see that there is a huge range of expression of GFP from the level that are still inside of the Q3 quarter, but also the ones that are already entering to the Q4. As we have been showing for the rest of the complex systems that are susceptible to exhibit SOC, we also plotted our data in these accumulated density plots. As you can see now, you have the GFP or RFP intensity in the x-axis in the log in a log fashion manner, and also in the log fashion manner, but you have the accumulated counts, how many particles. It's easy to see that the green line inside of the gray square is a line that it's showing a power law. And again, we calculated the exponential, it's around 2. And well, this actually is the demonstration that we succeeded in creating a circuit that was exhibiting self-organized criticality. And if you want to see the comparison, you can compare the, well, it's easier if you go to the panel, the upper panel, where we are showing the counts without the accumulation, and you can see the GFP intensity of a sample that doesn't have the SOC activated, like in green, or but is in the congested phase. And in the lower panel, you can see also the non-accumulated counts of the culture that doesn't have the congested machinery here in the blue dots. In comparison with the power law that the system exhibits once you trigger and you start the SOC motif by the addition of the correct amount of arabinose. And well, just to see that I've been presenting just one experiment with be performed that many times, and all of the experiments reproduce robustly this SOC behavior. And of course, now we are brightly looking into the future. we thought about doing that with a E. coli that doesn't have Lotka in their own genome, to see if we can reach another parameter space that exhibits SOC inside of using the same circuit. We also thought about designs that implies multicellular design to provoke this kind of SOC avalanches, but in a population. More interestingly, we are translating that circuits to be used inside of mammalian cells, because the mammalian cells have a cell cycle that's much longer than the E. coli, and then this will allow us to see if the SOC behavior also happens in the time scale. Also, we know that we have different sequences that allow for different ratios of degradation and ratios of saturation of the machinery. So this will be also something that will be good to explore, at least at the level of the model, and maybe thinking about as the first experiment that was proposed by Perback and with the sand piles, to see if the neighboring and the 3D lattices also do something like that. But at the end, we already engineered an stable attractor in living cells, and we think that this can be useful for generating phenotypic diversity in a bacterial population, or even to be linked to a drug delivery system to avoid adaptation of whatever you are trying to kill. For example, a bacterial lysis against colorectal tumors that has been already proposed in some works. So thank you very much for your attention. This is our small wet lab in the PRBB, the CSL, the Complex Systems Wet Lab. And thank you again to my coworkers, Ricard Soler, Blai Vidiella, Jordi Pla, Victor Maul, and more importantly also the technician that helps us with all this wet lab work, Ariana Rivera. And I think that with that I can... No.
Okay.
I know.
Thank you so much, Nuria. Very interesting talk. So, do we have any question while Nuria is recovering herself? Okay.
I'm sorry, it's the allergy.
Okay. So one question from Daniela Moore. Thanks, Nuria, really interesting. He says, if I understood correctly, the probability distributions are a snapshot of the state of many cells. Would single cells change states over time when in the critical state? Did you see this in the lab?
Before handling these questions to the ones that actually did a lot of work with the model, I said that I hope in bacterial cells. That's why I translating that system to mammalian cells, which we can actually do a video and demonstrate that. But we did several attempts to do a video with a SOC inside of bacteria, and looks like it is the case. But since bacteria are dividing in a cell cycles that are that are shorter than the fluctuations of SOC, it's very difficult to quantify and follow the bacteria. So we took advantage of the demonstration that the SOC, if a system exhibits SOC, you can see the power law. And we got this flux machine, the cytometer machine, and that's why we go in that direction. But I don't know if the people who was working with the model has a better answer for that.
Okay, so another question by Josep Mercadal. Do you know of any genes in bacteria or in eukaryotes whose activity self-organize in a critical state? Then if so, would that confer some interesting property, for example, coping with fluctuating environments?
Directly in SOC behavior, I'm not aware of, but something similar that some enzymes actually do the saturation and behaves in a kind of a Q waiting system. I don't know the name in English, but this has been demonstrated for some enzymes that actually collapsed and then decollapsed. We don't know exactly why, but this happens and has been demonstrated. But I don't think that anyone is actually looking for that. It's very difficult to see if this is happening inside of the cell, at least for bacteria.
Okay. He was also asking another question, which I think it's very well discussed in the papers. So Joseph, if you can stay alert for the release of this paper. But he was mainly commenting about the advantages of having these criticality states in terms of coping with fluctuations, for example, fluctuating environment. So I understand that he's asking if it would provide some kind of selective advantage and robustness to fluctuations.
Yes, yes, it does. It does give you an advantage to be in this kind of critical state, because then you can change. If you need to respond faster to something, that's for the first thing is that this gives you a phenotypic, this gives you a range of phenotypic if you have a protein in this range, in SOC behaviors, sorry, then it's probably you can see in a population level, you will have different concentrations of this protein. If then this population is exposed to some insult, then many of them or some of them will survive. So it gives you an advantage in that sense, because you are covering a range of concentrations of that protein in terms, if you think in a population level. And if you are not thinking in a population level and you want to think inside of the cell, to be in that critical state will allow you that with a few and a very tiny change in one parameter, allow you to respond in a very different way, fastly and easily.
Okay, another question by Patrick. Could you explain how you expect SOC to help resist adaptation? I would imagine delay between drug delivery rather than constitutive production would make a difference, but not necessarily the burst you get from SOC.
Well, I think that what we propose is that the unpredictability of the space between the bursts of the, imagine that this is a bacteria that's releasing the lysating, the lysate for the tumor cells. The tumor cell will not have a way to adapt, because will be impossible for the tumor cell to know when the next burst will occur and which will be the concentration of this lysating protein in terms of the size of the event. I don't know if this answers the question, or if any of the other authors want to add something.
Yeah, okay. So you're getting past the memory aspects of the cells. Yeah, okay, that makes sense. Cool.
Thanks.
Thank you.
Okay. Okay, so we want to thank the effort of Nuria of doing the talk. Thanks so much. Don't worry.
I'm not ill, it's just allergy.
Okay, okay. And so with Nuria's talk, we finished today's session on systems and synthetic biology and dynamical systems. So we want to thank all the speakers that we had really, really amazing talks and a lot of examples of combining mathematical modeling and dynamical systems theory with experimental research. So we are looking forward to see all of you tomorrow. Tomorrow there will be only a morning session about cancer systems biology, and we will start as every day at 10 a.m. So thanks so much for attending to today's conferences, and see you tomorrow.